Okay, some reminders for today. The quiz is open until 3 p.m. today. Last time I checked at 7:20, 34 people have done it, so I mean 26 of you still need to do it, although that was changed. Chapter 10 homework is going to be due on next Friday, which you're not thinking about yet, so it's great. I get it. It's not posted yet, so you can't think about it anyway. So I will post it soon, making no promises. And then the chapter 9 test will be on Wednesday, 10:23. Dr. Dietrich and I talked about it, and she is moving her test to Friday. So you will not have Koji County by the same day because we decided that was a great idea. Okay, so bio test is staying on Wednesday. Chem test will be on that Friday, 10:25. And happy Thanksgiving to all Canadians. I don't know if there's more than one in here, but there's at least one Canadian. So reviewing from last class, this is a quick review to get us back into cellular respiration, which we're going to finish today. This is foundational understanding. I know it's new before break, but it's foundational understanding for the way science biology works. So you need to be here today for 48 more minutes, and I don't care what you do, you can turn off your brain after that. That's I don't know if you have other classes. Okay, so thinking about cellular respiration, take a minute to answer these questions. About what's happening in the different steps. Pull out your yellow sheet of paper. We're gonna finish that today. I think I do in my office. I will go grab one while people answer these questions. No, they must Okay, when I come back, you need to answer all these questions right. I love this.
No, there's like
Does anyone else need a yellow sheet? Okay, so thinking about cellular respiration. What step or steps splits glucose? This is an easy one to warm you up. Glycolysis, yes, because it tells you in the name. What step or steps does not make any ATP? Which one? Pyruvate oxidation does not make any ATP. Which step or steps fills empty NAD plus electron carriers? The citric acid cycle does. Brayden, you want to add something? Pay attention. What other steps fill NAD plus carriers? Glycolysis does. Anything else? Pyruvate oxidation. So all three of the steps we've talked about so far do. Which step uses ATP? Glycolysis specifically, which part of glycolysis?
Investment.
There's two phases. There's the energy investment, which is steps 1 through 5, and the energy payoff, which is steps 6 through 10. So energy investment is the first 5 steps of glycolysis. What step fills the most electron carriers? Citric acid cycle filled how many NADs? We got 6 NADs out of that and 2 FAD H2s. Which step or steps takes place in the mitochondria? Pyruvate oxidation, kind of, as we're moving in while that happens, pyruvate oxidation and citric acid cycle, and the last one we haven't talked about yet, but yes. And which step fills FAD electron carriers? Yes, the citric acid cycle. Okay, so that's where we ended. At the end of where we ended on Wednesday, how many ATP molecules did we make? Look at your chart. How many have we made so far? We made 4, right? So we had how many from the first step? We made 2 nets. Remember, how many did we really make? We made 4, but we used 2, so we only get 2 out of that. How about pyruvate oxidation? We already know we made how many? 0. Citric acid cycle, how many did we make? 2, 1 per turn. Every time we go around the citric acid cycle, we make 1 ATP. So this is all we have in terms of ATP. Karen had finished earlier. I could have gotten this all up before then. Okay, we filled 2 NADHs in glycolysis. We filled 2 NADHs in pyruvate oxidation. And then we filled 6 NADHs in the citric acid cycle. And I print a new one so that I don't have a mistake again. I need a Vanna White or something. And we filled, last but not least, 2 FADH2s. We've done a lot of work, and we have very little to show for it in terms of ATP. We're getting there. So the last step, step 4, is oxidative phosphorylation. Oxidative tells us what's happening. Electrons are being lost because oil rig. Oxidation is lost. So we are losing electrons and we are phosphorylating, which means we are doing what? We're adding phosphate group. We're taking ADP and we're making ATP, which is the point of this reaction. Okay, this is going to happen in 2 steps because we can't make it simple. The first part of oxidative phosphorylation is the electron transport chain. And the second part is chemiosmosis. So we always call the electron transport chain the ETC because it's shorter. Okay, so we're going to talk through a little bit about what the electron transport chain is before we talk about how it works. You can probably guess what's happening in the electron transport chain. What are we doing? We are transporting electrons. Okay, that's a good easy one for today. Where is the electron transport chain located? Where are we now? We're in the mitochondria. Remember, the mitochondria has how many membranes? Two. It's got the outer membrane and the inner membrane, which is this folded thing on the inside. We're in this inner membrane. So this phospholipid bilayer, wow, is the inner membrane of the mitochondria. So the electron transport chain is these series of proteins, mostly, that are embedded in the phospholipid bilayer that is the inner membrane of the mitochondria. There's two sides to this membrane. This side is the side that's between the membranes, called the, it's between the membranes, so we call it the intermembrane space. And what's the space on the very inside called? We're going to the matrix. Okay, so this is the membrane in between the intermembrane space and the matrix. And again, we're talking about these series of proteins that are embedded in that. We have two different kinds of carriers of the electrons. Some of them are mobile carriers. Mobile meaning what? They can move. Okay, there's two mobile carriers involved in the electron transport chain. One is ubiquinone, which is we abbreviate Q, which is this one here. And then cytochrome C, which is here. The other four are just protein complexes. I don't know why I thought that we should write them all out, but we're going to, I guess. So complex 1 is an enzyme called NADH dehydrogenase. How do we know it's an enzyme? Enzinase. What do you think it's going to do? Based on its name. Its name is NADH dehydrogenase. It's going to take hydrogens off NADH, which also is going to move electrons too. That's complex 1. Complex 2, you can see down here. So these complexes 1, 2, and 4. 1, 2, and 4 are embedded proteins, so they're integral membrane proteins. Complex 2 is a peripheral protein. When we talked about membrane structure, we use those terms. So complex 2 is a is a peripheral protein. That one is succinate dehydrogenase. Again, I don't know why I'm making it right there. I don't think you need to know the names of them. It's not bad for you to know the names of them, but I'm not going to ask you to memorize them. And then we have the 2, 3, and 4 are both cytochrome protein complexes. Cytochrome B to C1 and cytochrome oxidase. Again, ACE telling us that they are enzymes, and they're all going to be involved in they have similar functions. Most of these are proteins. Some of them have embedded extra things added to them, cofactors or coenzymes that are added to them that are called prosthetic groups. And what's going to happen is the electrons, we're going to bring in some NADH, and we're going to strip off the electrons, and those electrons are going to move
down from 1 to Q to 2 to 3 to cytochrome C to 4. They're going to move down until at the end they are ultimately accepted by, because they have to go somewhere. So we said the overall equation of cellular respiration takes glucose plus oxygen. And we talked about the glucose carbons, and all of these glucose carbons are actually gone now, because where did they turn into? They're all released as carbon dioxide, but we haven't even talked about oxygen and how oxygen is involved, even though it's combining there obviously with oxygen too. But the oxygen that comes in is used here. This is the point of the oxygen. At the end of the electron transport chain, it is going to accept those electrons. So the electrons are going to move down this chain, and as they move down the chain, they are moving from an atom, from a molecule that is less electronegative. That says electronegative. I feel like we need to try that again. So they're going to move from an atom, from a molecule that is less electronegative to ones that are more electronegative. So it's exergonic because we're moving from less to higher electronegativity. It's like they're pulling them down. You can think of it like magnets. Like each one is a stronger and stronger magnet to be able to pull those electrons down the electron transport chain. All of these molecules, oxygen is the most electronegative. So it's going to pull the electrons down to itself. So oxygen is the most electronegative, and the NADH dehydrogenase is the least electronegative. And again, these electrons originally came from that glucose molecule. All carbon is gone, but these electrons that are moving down there are ones that originally came from that glucose molecule. So what does that look like in terms of what's going in, what's going out, and how is it working? So we're starting with NADH, full or empty? Is it a full or an empty electron carrier? It's a full electron carrier. So in a process that we're not going to go into all the details of, it's going to give away its electrons. So when it loses electrons, when it's in its NADH form, it's reduced because it has the electrons in it. It's reduced because reduction is gain. So the NAD plus has lost its electrons, so it is the opposite of reduced is oxidized. So this is the oxidized form. So we're taking the reduced form, turning it into the oxidized form, and the electron is going to travel. So it's going to travel from 1 to this mobile carrier Q. This color isn't showing up. So it travels following the yellow arrows. It's going to go from 1 to the mobile carrier Q, which is going to transfer to 3. 3 is going to transfer to cytochrome C, and cytochrome C is going to transfer to 4. Over here, you'll see with 2, which is our peripheral protein carrier, this is where FAD is going to come in. So this is FADH2. Is it full or empty? It's full. It's carrying the electrons. So it's going to lose its electrons. So when it's full, it's reduced. And it's going to become FAD, which is the oxidized form. So its electrons are also going to go to ubiquinone, which are then going to move to complex 3, cytochrome C, complex 4. Okay, so they're just going to bounce down this chain, be moved down the chain. Again, this is exergonic, which means we don't need to invest energy. It's just happening. But that's not the only important thing that's happening. It's not just that the electrons are moving, and at the end, we're going to take some hydrogen ions, because this is an aqueous solution in the matrix. We're going to add some oxygen, and we're going to make some water. So these electrons at the end are going to combine with hydrogen ions, which have a positive charge, some oxygen, so we get the neutral water product. And that's the water that's in the equation, the original equation. So this oxygen and this water are the ones that are in your original equation. But that's not the only thing that's happening. As each electron is moved, the movement of the electron actually provides energy to take a hydrogen ion and pump it through. So it's going to move through the matrix, out of the matrix into the intermembrane space through that inner membrane. So as it moves, it pumps a hydrogen ion. It's going to keep moving. As it moves into complex 3, another hydrogen ion is going to be pumped through. As it moves to complex 4, another hydrogen ion is going to be pumped through. So for each electron that moves, you also get one hydrogen ion pumped into that intermembrane space. So every electron equals one hydrogen ion moving through. So if we get a bunch of hydrogen ions over here, as we move a bunch of electrons through, because we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 NADHs that can give 2 electrons each, plus 2 FAD2s that give 2 electrons each. So we have 24 electrons that we've made that can move through this at this point. So that means we can pump 24 hydrogen ions through the gradient. What's through the membrane? What's going to happen up here when we have a high concentration of hydrogen ions? And over here we have a low concentration. What have we made? When we have a high concentration in one place and a low concentration in another, we've created something. Think about diffusion, osmosis. How do things move? From high to low through what we call the concentration gradient. So what we've done is we've made a concentration gradient. We've made a hydrogen ion concentration gradient. We have a high concentration in the inner membrane space. So we have a high concentration of hydrogen ions in the inner membrane space. So we've created a concentration gradient. And that's really important because that concentration gradient is a representation of potential energy. If you think about, you put a dam in the river and you put a whole bunch of water in there, that water has a lot of potential energy. And if you release the dam, it's going to come out. Okay, so we can think of the inner membrane as a dam, and we've created a whole pile of water behind it, which is just hydrogen ions, but that is a whole bunch of potential energy that's going to come into play as we move from the electron transport chain to the last phase, the chemiosmosis part of oxidative phosphorylation. So I don't think that yet. I want to, okay. I don't want to, we're not going to do the in and out until we get through the second part. Again, I just want to make sure that we understand that the point of oxygen here, I didn't write it down, is it's the final electron acceptor. Okay, that's the role that oxygen has. That's why you're breathing oxygen. You're breathing oxygen to accept electrons at the end of the electron transport chain during cellular respiration. And it does that again because of its high electronegativity. Okay, so at the end of this, what do we have in terms of electron carriers? We have full or empty electron carriers at the end of this part. We've emptied all of them. So all these ones that were full are now gone. Where can they go now? They can go back to the beginning and they can get filled up again.
So this is a recycling process too. They're empty, they can go get full again. So we've emptied all our NAD pluses. Oh, no, it should be 10, sorry. No, I can't count still. For 10, there's 10.
Yeah, I'm messing up.
What?
Sorry.
There are 10? I didn't break it, can't count to 10. I'll take that into my office. So now we have a whole bunch of empty electron carriers. And really, I should take down all the full ones because we don't have any more full ones, but that's okay. We're gonna pretend that we're running this simultaneously. Okay. So now we get to move into the final process, which is chemiosmosis. This is still part of oxidative phosphorylation. So oxidative phosphorylation has two parts. It has the electron transfer chain and then it has chemiosmosis. So we're still in the same thing and we'll do the in and out for the whole thing at the end of this. So chemiosmosis. What do we mean by osmosis in general? Osmosis usually refers to water. We're not talking in this case about water, but we're talking about hydrogen ions. And it's osmosis, so we're moving from high to low concentration. So we have a high hydrogen ion concentration, and we're going to move to a low hydrogen ion concentration until we end up with a equal concentration of hydrogen ions. Just like we would expect with water. If we have a high concentration of water, it's going to move through the membrane until it's equaled out. That's what's going to happen with these hydrogen ions. We have a lot of hydrogen ions in intermembrane space. And we have a low concentration of hydrogen ions in the matrix. So we're at the very last step and we have this weird looking thing. This weird looking protein complex is ATP synthase. What does that tell us? ACE tells us it's an enzyme. ATP synthase, what is it gonna do for us? It's gonna make ATP. Okay, it's gonna do that using this hydrogen ion concentration. Again, that high potential energy, we can take these hydrogen ions and they're actually gonna fill in this top part. There's little spaces for them. They're gonna fill it in and they're going to use that, it's gonna provide mechanical turning energy, like a water wheel. Okay, so we've built up our dam of water and now it's gonna go through the water wheel. And that water wheel, the movement of that is gonna provide the energy to take an ADP and add a phosphate molecule to it so that we have ATP. So this H plus concentration gradient provides what we call proton motive force. So it's going to turn the rotor of the ATP synthase. And that energy will phosphorylate ADP, adding that inorganic phosphate, making that bond. That energy provides the energy needed to make the bond between ADP and that inorganic phosphate. ATP synthase is an enzyme. It's got 4 parts of the protein complex. It is the smallest molecular motor, rotary motor that we know of. It's adorable. And it does really important things. Okay, how much does it do? Okay, so far, how much ATP have we made? 4. Okay, and I said it's a lot of work and we haven't really gotten much out of it. This is where the payoff happens. So this is like the big ATP synthesis here. This is going to make 26 to 28 ATP molecules. I did not bring 26 to 28 ATP molecules to hang up.
Because I don't have a Vanna White to do it for me. I just wrote 26 and 28. There's a little bit of a range because it depends on some different factors. The ratio of NADH to ATP molecules is not even. So one NADH will make 2.5 ATPs. FAD comes in a little bit later, so it doesn't make as much. It can only make 1.5 ATPs. And that hydrogen gradient, that proton motive force, is sometimes used for other things, like bringing the acetyl-CoA in to the mitochondria. So that force, not all the force of the proton motive force is used for making ATP, so there's a little bit of a range. But we're still talking about a significant number, way more than we made before. This is about a 34% efficient process. So about 34% of the energy in glucose is actually used to make ATP. 34 is a pretty high efficiency process. It doesn't sound very efficient, but it's actually a pretty efficient process. So let's talk about our final ins and outs for the step of oxidative phosphorylation. So on your charts, we already talked about where it happens. What is going in? What are we taking into the oxidative phosphorylation stage? We have 10 NADHs, which again are full electron carriers. That's what we've brought in from all the steps. So everything, all the NADHs that we made in glycolysis, pyruvate oxidation, and the citric acid cycle are all ending up here in oxidative phosphorylation. What do we get out of those? We had 10 full ones. Now we have 10 empty ones. Okay, what else? Now have 2 empty ones, so what did we have to bring in? We brought in our 2 full ones from the citric acid cycle. What else? ATP, which is what we want. Should say 26 to 28 ATP molecules. That's what we made. We did not use that, we made that. Again, thinking of both parts of this together, we put oxygen in. What did oxygen become in this reaction? It becomes water because it's taking those electrons and adding some hydrogen ions to it. So oxygen became water. These guys can all go back to the beginning. This can go towards cellular work, and water just hangs out because it's not really doing anything harmful. And that is how you make energy. Simple, clear, right? Okay, I'm not going to show you the video because I want to make sure we get through the very last thing, which is what happens if you can't do respiration. What if there's no oxygen available? But these are links, and there are links on Moodle also for the virtual cell videos that show this kind of in action. They show you how this turns, another hydrogen ion comes in, turns, another hydrogen ion comes in and turns, and that's going to produce that energy, that mechanical energy that produces the energy that's needed to make the bond between ADP and PI. So watch those at least before the test. We're going to skip the summary. We'll come back to it if we have time. Okay. We talked about all that happening in the membranes of the mitochondria. What happens if you're a prokaryotic cell? You have mitochondria? No, because there's no membrane-bound organelles. So for them, that same process can still happen. Prokaryotic cells can, some of them, still do aerobic respiration. It's just taking place
with the plasma membrane itself. So the gradient is happening like outside the cell versus inside the cell. So it still happens in prokaryotic cells, but not all prokaryotic cells can do aerobic respiration, and sometimes cells don't have access to oxygen. If you've ever been exercising and you're breathing some kind of aerobic exercise and you're breathing really hard, and you feel like you can't get enough oxygen, that's because you can't get enough oxygen. Okay, your body, especially your muscle cells, are calling for more oxygen and you can't get enough in there. So it's a desperation thing for a muscle cell to say, I need more energy. So there are alternative pathways. There's two alternative pathways to needing oxygen. These are anaerobic. So aerobic means using oxygen. Anaerobic, we're not using oxygen. The first one we're not going to dwell on too much. There is a process that is still a respiration process. It's still called cellular respiration, but it's anaerobic cellular respiration. So all of this is aerobic cellular respiration because it's involving oxygen. There is an anaerobic respiration. What did we say the point of oxygen was in aerobic respiration? What is it doing? At the end of the electron transport chain, it turns into water because it's accepting the electrons. So its job is to be the final electron acceptor at the end of the electron transport chain. In anaerobic cellular respiration, they just use an alternate molecule as an electron acceptor at the end of the electron transport chain. So there's still an electron transport chain. It's not that there's no electron transport chain. There's still an electron transport chain, but in the end, it's not oxygen that's pulling those electrons through the electron transport chain. It might be a sulfur or sulfide group pulling it through. So instead of making water, it makes hydrogen sulfide. If you've ever been to like a swamp, you smell that rotten egg smell. That's because there are bacteria there that are carrying out anaerobic cellular respiration and they're producing sulfide, hydrogen sulfide, which smells like rotten eggs. Okay, so anaerobic fermentation still has an electron transport chain, but no oxygen. The other one that we are probably more familiar with is the process of fermentation. The fermentation, again, is an anaerobic process. It doesn't use oxygen. It also does not use an electron transport chain. So this is no electron transport chain and no oxygen either. Both of them are anaerobic. So this is basically just going through the glycolysis process. Taking the glucose molecule, it's breaking it down into pyruvate. And there's two forms of this, which you can see here. There's alcohol fermentation, and there's lactic acid fermentation. So again, same thing, we're starting with glucose. So in both kinds of fermentation, we start with glucose and we get pyruvate out of it. And then it depends on what enzymes are present. So some organisms go through alcoholic fermentation. So they take this pyruvate, they process it into this acetaldehyde, which is then processed into ethanol. Again, we're going to have NADHs and NAD pluses cycling through where they're emptying and filling with electrons throughout this process. And we're going to make 2 ATP molecules. Instead of water, we're going to make ethanol. So this happens in some kinds of bacteria. It happens in yeast. And again, these electrons still have to go somewhere. So this, in this case, this acetaldehyde is the electron acceptor. And then it's processed by an enzyme to be ethanol. So this is how you've probably experienced fermentation reactions at some point in your life. If you have eaten bread, okay, that's a fermentation reaction. Alcoholic beverages, cider, We made cider last weekend at my sister-in-law's house and didn't have enough refrigerator space for it, so we're creating a low fermentation reaction on our front porch, which could have explosive consequences. So this pyruvate, again, just gets processed into ethanol ultimately for alcoholic fermentation, or it can be processed into lactic acid. In this case, pyruvate acts as the final electron acceptor. We're still making 2 ATPs. We're still taking NADHs and NADs and filling in and emptying electron carriers. But this pyruvate is processed enzymatically. Different enzymes make it into lactate or lactic acid. And again, you're probably familiar with this if you've ever eaten a pickle or kimchi or sauerkraut. Sourdough bread is processed instead of by yeast, it's lactic acid bacteria that are producing that sour flavor, and some yeast too. It's actually both kinds. And muscles. If you've ever been exercising aerobically and you're like, oh, I really have this burning feeling in my muscles, that burning feeling is the lactic acid that your cells are producing because they are desperate for energy. They're desperate for ATP. And because they don't have oxygen, the alternative process is to make lactic acid. And that lactic acid creates the burn that you feel. So it can happen in bacteria, and it can happen in muscle cells. And again, we found ways to harness these things and useful ways to find things that we like. What is the disadvantage of this for a cell? If we're talking about glucose, going through the process of glycolysis, making pyruvate, if we head this way where we have ethanol or lactic acid fermentation, it's a very short process, but what do you get out of it at the end? At the end of glycolysis, what do you have? How much ATP? 2. Okay, so it only produces 2 ATP. It's not a high energy process. So for if you're a bacterial cell, you're tiny, you don't need that much energy. But if you're talking about moving your whole body with muscle cells, you do need more energy. So this is an emergency procedure for you. For some bacteria, this is for some organisms, this is like a life thing and it doesn't bother them. But for you, this is an emergency. This would be considered, we don't know what to do. We have no other way to get energy, so we have to do this. The other way, when we make the acetyl-CoA, we oxidize that pyruvate and we go through the citric acid cycle. Again, we're getting 30 to 32 ATP total. So there's obvious advantages to this. But if you're a little bacteria, it's a lot of work to go through this whole process. You have to make a lot more proteins, you have to make a lot more enzymes, so it's a more complicated process. So some organisms are what we call obligate anaerobes, where they never use it. Some organisms are what we call obligate aerobes, where they can only use oxygen. And then there's some that fall in between. Mainly what we call facultative anaerobes, meaning they would rather do aerobic respiration, but facultative, meaning if the situation dictates it, they could do anaerobic respiration. Almost the last one. Okay, we talked about this as this is what happens when you have glucose, and this is how we do it all with glucose, but we don't eat a diet exclusively glucose, I hope. I hope your diet is not exclusively glucose. We've talked about the fact that we eat other things, and we know the monomers of these different things. Okay, whenever you take in anything, your body's going to digest it into smaller particles. So proteins are broken down into amino acids, carbohydrates are broken down into monosaccharides, fats are broken down into glycerol and fatty acids. All of these can enter this at any point depending on
Okay, when you break down proteins for amino acids, most amino acids are gonna be used to do what in your body? Making more proteins. But some of them can be, you can take off the amino group, it's called deamination. I don't know what that word. So we remove the amino group. That produces the byproduct NH3, which is urea, which makes urine. But then once that amino group is removed, it can enter at the pyruvate phase at the end of glycolysis. It can enter here, it can enter here and get to the oxidative phosphorylation phase. And again, same thing with carbohydrates, depending on their configuration, they can enter at different points. The glycerol molecule can enter here in the middle of glycolysis, and fatty acids can enter here at the acetyl-CoA stage and go through this whole process to be used to make energy. So it's not just that we take glucose and do all this. There are alternative ways to get through all of those. Your body also has to be able to control the process. Needs to be able to prevent waste from happening and preventing excess. So again, here's glycolysis, this is the citric acid cycle, this is oxidative phosphorylation. Your body can use different things kind of as a switch to turn on or off the process. So there are different molecules that can bind to this enzyme, phosphofructokinase, which can be bound to allosterically. And you can have inhibitors or, what's the word? Activators, sorry. So there's a molecule called AMP, which will actually stimulate. So it's like an activator. It will bind to the, we call it PFK molecule, and it will change the conformation so that it's more receptive, so that it will activate the process, so that it will happen faster. Citrate, which is one of the carbon molecules involved in the citric acid cycle. If you get too much of it, it actually will inhibit. And ATP also can be an inhibitor. So both of these can act as inhibitors to slow down cellular respiration. Or slow down glycolysis, because we're talking about how it acts just on that. So there are feedback mechanisms to prevent things from going crazy, from making too much ATP, for having things happen too fast, because your body needs to control that. If you're a hibernating mammal, you don't want too much cellular respiration going on, you don't need that energy. You don't want to break down your stores too fast. Okay. 1 minute early. Have a good break. Thanks. I looked at my stack and I was like, this is all I have to do during break. This is great.